Hey guys, I hope everyone is well. We are going to continue our discussion of genetics and inheritance. So far, we've talked about basic inheritance as described by Gregor Mendel, where inheritance is pretty straightforward, where the dominant masks the recessive, and we end up with dominant being expressed if we have a heterozygous genotype. So that would be like a capital T, lowercase t, and the plant is tall. Now we're going to look at other types of inheritance. Notice that complete dominance is what Mendel describes. That's often called Mendelian inheritance. Again, that heterozygous genotype produces a phenotype where the dominant masks the recessive and is expressed without mixing with the recessive. So we kind of hide that recessive, so the heightened P plants. But if you think of most human traits, or a lot of traits that we might think of with other organisms, they don't follow this complete dominance where the dominant completely masks the recessive. It's not that straightforward. So you have a lot of other patterns of inheritance or types of inheritance that won't be demonstrated by what Mendel studied. And these are kind of like exceptions to the rule. So you do have a chart that you can fill out. It's not really an outline. It's more just a chart that would allow you to record descriptions as well as examples of these different patterns of inheritance. So the first one is called incomplete dominance. So instead of having a dominant masking a recessive, we now have the two alleles that will mix. So one allele is not dominant over the other. Instead, if we have a combination like in the heterozygous genotype, we end up with a mixed phenotype. So the two kind of give us an intermediate phenotype in the middle of the two extremes. So maybe we would have a heterozygous pink flowering plant versus a red or white flowering plant. And let's look at some examples of that. So flower color, here would be an example of that pink, white, and red flower. Notice the homozygous. So in this case, it looks like it's homozygous dominant, even though one is not dominant over the other. Notice the homozygous red versus the homozygous white. So if we have the other allele, which in this case looks recessive, then that flower would be white. But if we have the heterozygous, notice the heterozygous gives us that kind of intermediate color or an in-between color, which might be considered a mixed color. So if we have a heterozygous plant, it's going to be pink in this case instead of red or white flowering. So those genotypes are going to give us an additional phenotype to account for. So here's if we look at kind of 3 generations of this. So generation 1, we have a red flowering plant that is homozygous. We cross that with a white flowering plant that is also homozygous, and we end up with the heterozygous individual. The heterozygous individual is going to express that intermediate or that mixed phenotype. Now in the F1 generation, we see all of the offspring are heterozygous because the red flowering plant passed down the red allele, the white flowering plant passed down the white allele. But if we take that F1 generation and we cross 2 pink flowering plants from that generation, we're going to end up with about a 25% homozygous red, a 50% heterozygous pink, and a 25% homozygous white. So this is seen in snapdragon flowers. Our next type of inheritance is called codominance. So take a look at these terms. The names of these patterns is pretty helpful. So if we think of co-owners of a store, for example, they're both owners. So codominance means we have more than one dominant. So 2 alleles are dominant. So remember your rule about dominance is that if the dominant allele is present, the dominant allele will be expressed. But it's not mixing like we saw in incomplete dominance. So the heterozygous genotype gives us a phenotype where there are 2 different alleles being expressed at the same time, and they're going to keep their own characteristics, so we don't have a mixing. So take a look at our chicken here. Notice the chicken has 2 different colors. We'll see that example in just a moment. Human blood types are a really good example of codominance. So in human blood type, A is dominant, but so is B. So A and B are both dominant, which means they are codominant. But they're not the only blood types that we will see. We'll have some examples of that in a little bit. So notice type A. To have type A blood means you have produced protein A. To have type B blood means you produce protein B. But if you inherit an A from one parent and a B from the other, you're going to have AB blood. A and B are both dominant, so they will both be expressed. Now there is an additional blood type, and that would be O, meaning you inherit neither A nor B, so you don't make either of those proteins on the surface of your red blood cells. So there's your AB blood type as a result of having both of those alleles. So in this case, we're going to be heterozygous, but codominant. Here's an example of codominance in flowers. So notice this flower has two colors. It's expressing the pink and the white. So the genotype would be considered heterozygous, showing both of those colors because both are dominant. They are codominant. Here's that codominance in chickens. So this would be sometimes referred to as a checkered chicken. It has black colored feathers and white colored feathers. Again, it would be heterozygous codominant, showing both black and white feathers, sometimes called a checkered chicken. Here's an example of codominance in cows. So this cow is a result of a white cow crossed with a brown cow to give us the brown and white colors showing, but not mixing. We don't have a tan cow, we have a brown and white cow. So this brown and white cow is referred to as a roan cow. So it would be the genotype here. It would be heterozygous codominant. All right, moving on to polygenic traits. Notice the term polygenic. Poly means many, genic refers to the gene. So in this case, now we have many genes. We don't just have one gene that controls the trait, like we might have in, say, height in pea plants or pea shape in pea plants. Now we have many genes, which means there might be a gene on chromosome 2 and another gene on chromosome 10 and another gene on chromosome 20. We could have hundreds of genes that might contribute to a trait. So that makes it pretty unique in terms of how we'll express these traits. So we have many genes, polygenic, contributing to the expression of that trait. The more genes that we have, the more variety we might have. So we're going to end up with a lot of different possible phenotypes. So if you think of a trait in humans where we have a huge variety, it's most likely going to be a result of a polygenic trait. What happens is we have kind of the additive effect of all of those alleles adding up. So if we have height, for example, in humans, and tall is dominant over short. So if you are very, very tall, then you have a lot of dominance sprinkled over those many, many genes. The more dominant you have, the taller that you are potentially able to be. Now, your nutrition does contribute to height as well. Human skin tone is another great example. Notice the huge variety of human skin tones that we could possibly have. It's an additive effect of a whole bunch of genes contributing to that particular trait. Here's an example in wildflowers. So think of a field of wildflowers. You might see many, many colors because wildflower color is contributed by many, many genes. So it's a polygenic trait. So all those genes are going to kind of work together to give us that expressed phenotype, which might give us a huge variety of expressed phenotypes. Polygenic inheritance, human height is another example. So notice the variety of heights. So think if we were to take a large group of people and divide them up by height, we're going to have some people that are very tall, some people that are very short, and then a whole bunch of different varieties of heights in between. So we might have more that are in the middle, but we still will have this huge variety because we have polygenic inheritance being shown here. We have lots of different possible phenotypes being expressed. Some other examples, think of like human hair or eye color, because of course they have lots of variety as well. But we're going to see eye color with a different pattern of inheritance as well. And that would be multiple alleles. So multiple alleles means that we have more than two possible alleles. So think back to, say, Mendel's studies with pea plants. We use the big T to represent the tall plant, so the dominant allele, and the recessive allele, or the lowercase t, represent the short plant. And that was it. Pea plants are either tall or short. There's nothing in the middle. We don't have lots of possible alleles. We don't have a polygenic trait. We don't have mixing. So notice in multiple alleles, we have more than two possible alleles. It's not just one dominant and one recessive. We might have two dominants and a recessive. We might have a dominant and multiple recessives. We have many possible genes. So whatever two you happen to get will determine your phenotype. So eye color is a good example. So eye color follows multiple alleles, but it also follows polygenic. So we're going to have a little variety here. So you can see we do have an increase in the number of possible phenotypes, because we do have more possible alleles in the population. So we have brown, which is dominant, but then we have green and blue, which are both recessive. So brown, green, and blue are three different possible alleles, not just two possible alleles. So eye color is controlled by multiple alleles. There's a brown allele, a green allele, and a blue allele. And also it's controlled by more than one gene. So we have both multiple alleles and polygenic. We can have traits that follow more than one pattern of inheritance, as long as it's logical. So we always get some questions about having two different color eyes, or maybe even an eye color where you have the majority of your eye is blue, but then you have a brown spot. So having two different eye colors or that spot in the eye is called heterochromia. So it's when you either have, a partially colored eye that's different a color than the rest of it, or two completely different eye colors, as you see here in this picture. So this is a result of melanin being unevenly distributed, which is similar to freckles. Freckles are due to uneven distribution of melanin. So melanin is what gives us the coloration in our skin, in our eyes, and things like that. So we have a mutation in some of the cells. So we might lack melanin completely in one of the eyes, or might have that uneven distribution. So there's lots of things that might cause this. It might be genetic, it might be due to an injury, it might be due to some sort of disease that causes those cells to no longer make a pigment or not be able to make a pigment. So we end up with different coloration in the eyes. Okay, here's another example of multiple alleles in blood types. We saw blood type as an example of codominance. Now we have blood type as an example in multiple alleles because we have more than two alleles. So A is an allele, B is an allele, but O is also an allele, which causes the absence of the A or B proteins. So there are three different alleles that code for human blood type: the A, the B, and the O. Right, so you'll notice here in this chart that you see letter I here. We're going to actually use I's to represent blood type, believe it or not, when we look at our Punnett squares. All right, pleiotropy. This one's a little tricky simply because of the name, or partially because of the name. So pleiotropy is when we have one gene. It's kind of like the opposite of polygenic. In this case, we have one gene, but we have lots of different possible effects. The way that it is expressed seems to impact multiple areas. So we're going to see this in what's called sickle cell anemia. So sickle cell anemia is when an individual makes sickled-shaped red blood cells instead of normal red blood cell shapes. So notice on the left here, these are normal red blood cells. You might remember these from earlier in the year, this shape, how we kind of have this disc shape that's really flexible and it's kind of sunken in the middle. And they flow very easily, and they can squish and slide through small places like blood vessels. Sickle cell anemia individuals produce sickled-shaped cells. These cluster and clump. They are not very efficient at moving through the bloodstream. And so we end up with a whole bunch of different impacts of this for an individual. So even though it's only one gene that causes those cells to be sickle in shape, we end up seeing this expressed in many areas. So weakness, anemia, brain damage, spleen damage. So notice the multiple impacts it has, even though it is only one gene that's causing that trait. Oh, and heart damage, excuse me. All right, sex-linked traits. So sex-linked traits are unique because they are not found on our autosomes. They are instead found on our sex chromosomes. This is going to impact us in a way that it's expressed more often in males than females. So we need to account for the sex chromosomes of the individual, not just the genotype. And why are they found more commonly in males than females? Well, think back to our sex chromosomes. Females have 2 X's, males only have 1. So males only need one recessive allele, usually sex-linked traits are recessive, in order to express the trait. Females need to have both recessives, and it's just less likely that we will inherit 2 recessives than 1. So females usually have a second dominant allele to mask that recessive allele that would cause this trait. So we're going to see Punnett squares that are a little unique because we now need to account for not just the genotype represented here by A's, but also the chromosomes that are associated. So our Punnett squares, like I said, we're going to do some unique Punnett squares as we go through all of these patterns of inheritance. So I'll show you how to set this up. So here's a couple of examples. Colorblindness is an example of a trait that is inherited by a sex-linked trait. So therefore it's found on the sex chromosomes. And here's just a kind of a fun little practice here, a little colorblindness test. So individuals who are colorblind have a difficult time distinguishing the colors, the different colored circles here. Individuals who have normal vision do not have difficulty distinguishing the colors here. So this would be specific to red-green colorblindness. So we can see if we are not colorblind that these circles have an orangish, maybe reddish coloration to them. So you can see the number in this case 45. Sometimes it's a letter or a shape, where the other